and by way of disclosure, my group is housed at BioMed X Institute in Heidelberg, Germany, and more recently in Connecticut in the United States. Our innovation model is to partner and collaborate with industry funders to identify high-risk, high-reward projects that might otherwise go unfunded, but which, if solved, would move the needle significantly in terms of the way that patients are treated. Now, in 2018, the problem presented to me as an incoming group leader was how to de-orphanize T-cell receptors in virtually any application. The highest hanging fruit actually being autoimmune disease, which is why we started developing a platform around CD4 TCRs and class 2 presented epitopes, realizing that if we could overcome this hurdle, we could backtrack to things like CD8 class 1 presentation fairly easily if the platform were flexible enough. But what we did want to do, although admittedly the clever solutions are out there, we wanted to stay as physiological as possible and not manipulate any of the molecular interactions at the exterior of the cells, but also not at the interior of the cells. And I'll talk about that in a second, because I think it's one of the things that allows us to investigate things like the dark proteome. We are not over-reliant on predictive models and can even be fully independent of them in terms of true de novo functional antigen identification. What came out of this at the end of 5 years of work with the team was a platform that we call FINDER. The work that I'm presenting today is all published, so I encourage you to have a look at the publications, because I won't have the time to show you all of the interesting data that we got along the way. But essentially, there are two key sort of what I would say novelty factors to the platform. The first one is that in 2018, we actually were not very impressed with the signal to noise ratio of the typical NFAT reporter, or the ability of CD69 to come up quickly enough in cellular assays. So we developed our own TCR inducible promoter. It has this 100-fold signal to noise ratio, 3 logs of linear range. It can really capture those truly low avidity TCRs in the 10 micromolar range in terms of functional reactivity. I also completely agree that we would never measure the biochemical affinity of a TCR for its ligand. This is all functional readout based on our cell-cell interaction assay, which contains also a genetically encoded open reading frame that is processed naturally by the antigen-presenting cells, which are autologous, and allows larger open reading frames to present the epitopes as a donor's cells naturally would, making this interaction here at the surface of the cell completely physiological. And that is actually for class 2 not necessarily given. We found out early on that sustained strong processing and presentation of class 2 epitopes is not automatic in our antigen presenting cell lines. But we really wanted those long open reading frames for multiple reasons. As you can see here, you can take a 400 amino acid sequence and get multiple overlapping epitopes and work down in a functional manner, so we save the cost of peptide libraries. Just doing a little back of the envelope calculation, with 50,400 mers, we can cover the entire reference human proteome. And then just in terms of single amino acid resolution tiling, we can move down once we have identified the let's say large mini gene in question. We can identify the minimal functional epitope here. And I'm giving a few examples of DR4 restricted epitopes. These are class 2 presented epitopes. And actually, interestingly enough, and you'll see this in the paper, this is a 17-mer. So we can, probably the minimal peptide length that's presented is much shorter, but in terms of functional genetic components, you do need a few additional amino acids. So we took a combination of immuno-oncology approach and the CD4 perspective and collaborated with Michael Platten in Heidelberg, who had been treating midline glioma patients. This has a median survival time of about 15 months, universally lethal. And they had been administering a histone 3 K27M neoepitope as a peptide vaccine, and had a patient who survived 3 years post-treatment. And I suppose the question on everyone's mind was, is this because the immune system was actually responding? And part of our platform's novelty was its speed. So within 2 weeks, we actually could provide this figure and for the publication identifying nearly 3 dozen neo-epitope reactive TCRs. Interestingly, the clonal abundance or frequency is not necessarily correlated with the functional reactive response. So we're not talking about the first 10 or first 20 clonotypes that we pulled out of the patient's peripheral blood and cerebrospinal fluid, but actually going all the way down to about 200 in terms of how many TCRs one should screen if one wants to completely catalog the functionally responding TCRs. This ranking is just ranked by functional response, showing that we can actually sort of pick from these if one wants maybe a more effective TCR, and I think we can discuss this over the lunch break. Maybe maximizing functional readout is not the best strategy. So these were all CD4-derived TCRs. They had a central memory phenotype, sort of suggesting that maybe we ought to look for help provided to B cells. And we did find a B cell clone in the CSF of this patient. And once we turned it into a secreted antibody, we found that it bound selectively and quite nicely actually to the mutant peptide, at least to the vaccine peptide. What an antibody in the cerebrospinal fluid would be doing as seen in intracellular epitope, we're not quite sure yet, but maybe it's functioning more as a BCR and internalizing destroyed cells during the response and keeping the tumor at bay. We also did this is a new phrase I learned today, paragenic HLA mapping, and showed that this peptide was being presented by quite a diverse array. A second donor actually had DP presented peptide. We were able to deconvolute a few TCRs from that patient as well, showing that actually this peptide can be used in approximately 60% of the patient population, which, given that it's fairly rare to begin with, was nice to see. Subsequently, we moved on because AI/ML is sort of the way of the future. Again, together with Michael's group, we worked together to sort of functionally validate, as we've seen earlier today, TCRs directly out of a patient against the tumor cell line, and use that to sort of reinforce and retrain a classifier model, which has about 82% ROC. So it's actually doing quite well in terms of taking TILs and evaluating which of those might be or will be tumor reactive if placed back into the patient as a gene therapy. So hoping for about a 6-week turnaround time therapeutically. And then finally, thinking about where else we can go from here. Due to the nature of the long mini genes, we are able to identify peptide epitopes that haven't been otherwise predicted because we are fully functional. What we haven't been able to do in the past is identify TCRs of interest without the help of collaborators. So we actually set up a second platform that allows us to do this with in vitro expansion. So it's quite sensitive to pull out from a single T cell or the TCR that's responding. For CD4 TCRs, this of course is essential because the repertoire in the peripheral blood is very complex and you don't see a lot of clonal expansion necessarily. So we did a proof of concept in COVID because that was what everyone was doing in 2021. And then we actually worked for a while on celiac disease, sticking to our original mandate of autoimmune disease. We're able to show actually nicely the cross-reactivity to bacterial epitopes, so sort of chasing that epitope mimicry hypothesis. And it's a bit of hand-waving, but we do call this post-translational modification, changing a Q to an E in the mini gene, because that mimics the deamidation process seen in celiac disease. Behind the scenes, we've been working on the higher hanging fruit, such as post-translationally modified targets, and the system, as you saw, it is all set up and is currently ongoing in development to put these into ultra high throughput microfluidic assays, so single cell passages of TCR repertoires versus putative target libraries. That's that we will also be able to functionally deconvolute thousands of interactions per donor, ideally. So just to run through a few of the highlights of the platform as presented. By the way, BioMed X Institute is sort of a commercial / academic hybrid. We are open to collaborations from all directions. If anyone is interested, I'm happy to discuss it after the talk or at the poster session. To our knowledge, it is sort of the best in class in terms of functional deconvolution. I think we could arm wrestle over whether who wins there, but I think without making that claim, I wouldn't be here today. And I think we're really focusing on that physiological interaction, the sensitivity of our reporter line, and finally the physiological nature of the processing and presentation on the donor side. I wouldn't be here today without my team and our collaborators, so I do want to briefly acknowledge them before maybe I have a second, 2 minutes actually, to take questions. Thank you.